A quantitative comparison of the cytochrome P450 transcript in 3 different model organisms.
Clotrimazole is an antifungal chemical that breaks down the cellular membrane of fungal cells, causing the leakage and the subsequent breakdown of nucleic acids, thus allowing for the destruction of fungal cells and explaining why clotrimazole can cause burning and irritation of the skin in some cases.
In addition to its effects on fungus, clotrimazole is a chemorepellent for Tetrahymena, most likely for the same destructive reasons as in its fungal effects. Clotrimazole interacts in most organisms with TRPA1 channels and cytochrome P450. Tetrahymena don't have TRPA1 channels or regions of their genome analogous to TRPA1 in other organisms.
Because of the tetrahymena's lack of TRPA1 channels, we wanted to investigate whether or not the reason for the tetrahymena's avoidance of clotrimazole might be an interaction of cytochrome P450 similar to that found in other organisms.
In order to do this, we decided to determine the similarities between the gene transcript of cytochrome P450 in Tetrahymena, mice, and humans. If cytochrome P450 is a part of the mechanism of action for clotrimazole in Tetrahymena, we would expect to see similarities between the cytochrome P450 gene transcripts found in humans and Tetrahymena.
To make the comparison of the cytochrome P450 gene transcripts between humans, mice, and Tetrahymena, we used 3 different websites that allowed us to find the genomes of the organisms and then match the gene sequences. Using the website ensemble.org, we were able to examine the cytochrome P450 transcript in 2 model organisms, humans and mice. To examine this genome sequence in Tetrahymena, we used the website ciliate.org. After obtaining the 3 transcript sequences, we used the European Bioinformatics Institute website to compare the transcripts and determine the number of matched genes between the 3 different organisms.
In our comparison, we found that humans and mice share 86% of the same genes on cytochrome P450, while humans and Tetrahymena share 52%, and mice and Tetrahymena share 54% of the same genes on cytochrome P450.
What our data tells us is that the mouse and human gene transcripts for cytochrome P450 are very similar, which is not surprising considering the frequency of rodent models in medical experimentation.
Our data also indicates that neither the mouse and Tetrahymena comparison nor the human and Tetrahymena comparison demonstrate high levels of similarity between the cytochrome P450 gene transcripts of the different organisms.
The similarities between the human and mouse transcripts indicate that cytochrome P450 most likely performs a similar function in both mice and humans. Since the cytochrome P450 is affected by clotrimazole in humans, it probably also functions the same way in mice.
The differences between the human gene transcript and the Tetrahymena transcript, as well as the differences between the mouse transcript and the Tetrahymena transcript, most likely indicate that cytochrome P450 is not analogous between the Tetrahymena and the other organisms. This probably is evidence that cytochrome P450 does not function the same in the Tetrahymena as it does in humans.
Since cytochrome P450 is involved in clotrimazole responses in humans, the lack of similarities between the transcripts of cytochrome P450 and Tetrahymena in other organisms most likely means that cytochrome P450 is not part of the clotrimazole response in Tetrahymena.